I'm John Morrison. I'm a professor in the Department of Neurology at UC Davis. My research interests have always centered on the neuronal and synaptic organization of the cerebral cortex. It has a certain twist to it in that it's related to the neuronal and synaptic changes that lead to age-related cognitive decline, and also what happens in the brain that leads to the dementia of Alzheimer's disease. We spent a lot of time on the difference between age-related cognitive decline and what happens with Alzheimer's. One of the things you think about when you first get elected president is, oh my God, what's my logo gonna look like? There's a team of artists. She reads my papers. She tries to get a sense of what's important to me and what's important to my research through the years. She ended up with the pyramidal cell itself, the dendrite, and spines. It's really the thin spines that are lost with aging. And the thin spines are the most plastic. So what's really happening with aging is, yes, you're losing synapses and you're losing spines, but you're losing plasticity. We have worked for many years, actually for decades now, on what happens to these key pyramidal cells in prefrontal cortex with aging that leads to cognitive decline. What she's shown here is the yellow is the young set of spines on a typical dendrite, and the blue is aging where you've lost the thin spines. And she used these pyramids to reflect that it happens in a pyramidal cell. My favorite thing I've done at SFN is that I led the neuroscience team for the Life of a Neuron exhibit that Arctic House put on. We delivered the neuroscience to them. I think we should do more of it. I think it's a fantastic educational tool because you can teach people a lot about neuroscience in a way that's very fun and that is linked to art and linked to aesthetics. The more we can take advantage of that, the more you're going to engage the public.
And then we'll just get an image of you holding this up, and you can just smile with it like it's your Oscar.
It is my Oscar. Yeah, it's the best.
Good afternoon. My name is Teresa DeRochers. I'm an associate professor in the Department of Neuroscience at Brown University and a member of the SFN Program Committee. First, just a few announcements before we begin. Please take a moment to silence all electronic devices. Remember that photos and other forms of recording are prohibited unless the green camera with a checkmark image is displayed. There will be a question and answer session at the end of the session, so please stay for that. Questions can only be submitted through the Q&A feature available in the session description of the meeting app or the virtual platform. You can ask questions at any time throughout the presentation and as many as you like. I will ask as many questions as possible during the Q&A after the presentation. If you have to leave during the Q&A, please do so as quietly as possible. The session is being recorded and will be available within 24 hours through December 21st. So now, it's my honor and great pleasure to introduce Dr. Michele A Basso, and I would like to think that they saved the absolute best for last. Thank you for being here at the end. She's a professor in the Department of Neurobiology and Biophysics in the School of Medicine at the University of Washington. She earned her bachelor's degree from Fairfield University and her master's and PhD at SUNY Stony Brook with Dr. Craig Evinger. After that, she completed her postdoc at the National Eye Institute with Dr. Bob Wertz. She was faculty at the University of Wisconsin and UCLA before her current position at the University of Washington. In her career, Dr. Basso has shaped our understanding of decision-making, specifically in subcortical circuits involving the superior colliculus and basal ganglia. She has shown that these structures are critical in flexible, goal-directed choices. I won't steal her thunder and describe more of that fascinating work now, but suffice to say, if you only think about the cortex in decision-making, Michele may persuade you to take a closer look at some of those deeper structures. In addition to her amazing science, which has garnered her awards too numerous to list through the years, Dr. Basso has also done a lot for the field. She served as an editor for several journals, served as a member and chair of committees for SFN. In short, she has been an amazing and seemingly tireless advocate for science. Please join me in welcoming Dr. Michele Basso.
Thank you so much, Teresa, for that really generous introduction, and to you and the program committee for inviting me. It's really an honor and a delight for me and the members of my lab to talk to you today and to share with you some of the work that we've done over the years. So I always start my talks with the work of Yarbus, who was a psychologist from Russia, who did some very interesting work. And he showed healthy participants, people, these images, and at the same time, he monitored eye movements while they looked about these images. And the lines here show you times when the person made a rapid movement of the eye, what we call a saccadic eye movement, to look at different parts of the scene. And the points here indicate times when the eye remains stationary in the orbit. Presumably the subject is acquiring visual information, making decisions about where to look next, and so on. And so what he showed was that when people looked at images like this, and he gave them a set of instructions like estimate the material circumstances of the family, he obtained a particular pattern of eye movements here. But when he gave a different set of instructions, estimate the ages of the people in the room, there was an entirely different pattern of eye movements that ensued. And what this tells us is that we can think about these saccadic or eye movements as kind of a window onto thought processing. And in this particular case, we can think of it as a behavioral assay of the set of instructions that were given to the participant. And it's really this basis on which the field of perceptual choice and decision-making has rested. Many of us use eye movements as a readout of the perceptual choices that subjects make or participants make. And so the other thing that you can see in Yarbus, which is why I like to use this example, is that there are many objects in this scene, but the eye can only move to one of those locations at a time. You can only make one saccadic eye movement at a time. So there must be a process internal to the participant, internal to our brains, that identifies individual objects, tags them either for further perceptual processing, such as attention, or as a goal for a movement, such as an eye movement. So this is a second reason why eye movements are so critical to understanding sort of cognitive processing. Now, this is a lateral schematic view of the rhesus macaque monkey brain. It's the primary participant subject that we use in our experiments. And what I'm highlighting here are the cerebrocortical areas that contain neurons that discharge action potentials in association with the generation of saccadic eye movements, and also the visual stimuli that guide those saccadic eye movements. And importantly for the talk today, these cortical areas have direct projections down to this midbrain structure called the superior colliculus, which in turn has direct projections down to the brainstem, structures without which you'd be unable to move your eyes. So one of the things that my laboratory and the members are interested in is understanding how the transformation takes place from cortical information through the superior colliculus to the brainstem to generate eye movements, this sensory motor transformation. And we're also interested in a set of subcortical structures called the basal ganglia, shown schematically here. And the important thing is that the basal ganglia receive virtually the same cortical information, not all the same, but as the superior colliculus. And two of the output structures of the basal ganglia project to other parts of the brain. And one in particular, the substantia nigra pars reticulata, has direct projections to the superior colliculus. And so one of the things that we're interested in from a basic science perspective is understanding what these two pathways, the cortical collicular pathway and the cortical basal ganglia colliculus pathway, contribute to that process of sensory motor transformation, perceptual choice, decision making. And it's not only important from a basic science standpoint, it's important from a clinical standpoint. So understanding what the basal ganglia do for behavior, the importance of it for understanding clinical phenomena really cannot be overstated. There are so many disorders, neuropsychiatric and neurological, that involve damage or some degenerative process within the basal ganglia, that understanding how it works is of vital clinical importance. So examples include, of course, Parkinson's disease, Huntington's disease, dystonia. On the neuropsychiatric side, there's Tourette's syndrome, together with cortex, autism, schizophrenia, drug addiction. So the clinical relevance of basal ganglia really cannot be overstated. Now, the superior colliculus is, and its non-mammalian homologue, the optic tectum, has been studied for at least a century or more. And what we know from all of this work is that it is a structure that is fairly well conserved throughout the vertebrate lineage. So you can see down here the lamprey. The superior colliculus here is quite large, shown in blue, and this dates back, the lamprey dates back to 550, 600 million years ago. So they're considered the stem vertebrate, and all of the vertebrates since that time also have a superior colliculus. So it's considered a highly, highly conserved structure. And you can see here in the bird that it's actually highly developed, in fact. And Eric Knudsen, who is a main contributor to understanding the colliculus optic tectum in birds, argues, and perhaps rightly so, that it is highly developed in birds because of the unique challenges that birds experience visually during flight. And so I also encourage you, if you're interested, to explore the work of Sten Grillner in the optic tectum of the lamprey. It's some of my most favorite neuroscience. Now, the cells are generally conserved in the superior colliculus, and you can see a couple of examples here. These cells look very similar, but they come from two different animals. The details are not so important, but what you can see is that they're conserved even in diurnal species as well as nocturnal species. But you see there's a lot more specialization here. So the cells are very similar across vertebrates, but they have different kinds of specializations depending on how much their brain depends on the superior colliculus for vision. So as we evolved, primates evolved, the vision, the nuts and bolts and the hard work of vision kind of transitioned to the thalamocortical circuits. So the superior colliculus has become less important. And you can see here that the laminar structure of the superior colliculus also reflects this. So in particular, the avian species, the bird English sparrow here, or the pigeon here, highly, highly specialized lamination within the superior colliculus, where optic tectum in this case. And the gray squirrel also, as well as the tree shrew, two mammals, that depend on their colliculus for vision. You can see it's specialized. Whereas in the macaque, you still have the layers, but they're not quite as specified as they are in these other species. So these maps, or layers rather, form maps of the world, both visual space in the case of visual animals, and this is just an example showing you in a mouse and a monkey that the arrangement of the map is very similar. So for visual stimuli that are close to the central gaze, the fovea, straight ahead or nasally, you can see that they're represented anteriorly in the colliculus, and those more peripherally are located in the posterior colliculus. And medial colliculus represents space in the upper visual field and lateral in the lower visual field. And what's fascinating about these maps is that if you look at species that don't have vision, like this star-nosed mole, this beautiful work by Ken Catania and his group, these animals are not visual, they barely have eyes, and what they have instead are these fleshy appendages around their snout. You can see them here. And if you number them, as Ken and his team did here, you can see that their colliculus maps in the same way, but for somatosensory space. So close to the nasalward, close to the fovea, the somatosensory fovea in this case, so 1 and 11, you see are represented rostrally in the colliculus. And as you move more peripherally, those are represented further posterior in the colliculus. So the maps are really quite fascinating, and they span all the animals in the vertebrate lineage. Now, in macaques, perhaps one of the best thing that is known is for the saccade-related activity, the eye movement-related activity in the superior colliculus. And this is just an example of a neuron recorded from the deeper layers, or intermediate and deep layers of the colliculus of a macaque. Each tick here is an action potential. Each row of ticks is an individual trial when the monkey made an eye movement. And these are the eye position traces here, plotted against time. And you can see this average activity. There's quite a robust discharge of action potentials associated with the generation of the eye movement. And this is perhaps the most well-known thing about the superior colliculus in the monkey. And some really famous, influential work done in the 70s showed that, in fact, in the intermediate and deep layers of the monkey, there is also a map of motor space. So if you electrically stimulate, this comes from David A. Robinson's work, if you electrically stimulate in these regions of the colliculus, you see that you can evoke eye movements that are similar to the overlying visual space. So in the rostral, you can evoke very small eye movements to foveate a stimulus. And in the posterior colliculus or caudal colliculus, you can evoke much larger eye movements. So there are these lovely maps, and in my view, there's rich evolutionary studies that can still be done using the novel technologies that we have now in molecular biology and genetics to understand the evolution of these maps. So one of the things that excited me many years ago was an observation made by Okihita Hikosaka and Bob Wirtz. And that result is illustrated here. So what they did in the 80s really was they recorded from neurons within the basal ganglia the substantia nigra pars reticulata, the structure that projects directly to the superior colliculus, and the neurons contain GABA, the inhibitory neurotransmitter. And what they showed, here are eye movements again. They had monkeys fixate on this spot and then make a saccadic eye movement to these targets here. And then they recorded the neuronal activity from the substantia nigra. And what you can see here is that these neurons have high tonic discharge rate. They discharge a lot of action potentials in time, about 75 to 150 spikes per second, actually. And they have this tiny little pause here, you can see, when the monkey made a saccadic eye movement to that visual target. But the same neuron now, if you have the animal make the same movement, but now the target is guided by memory, you see that this neuron has profound modulation. And this was really one of the first demonstrations that the basal ganglia might be involved in covert processes like memory. And so it also fit at the time with the thinking about how the basal ganglia worked, that the basal ganglia participated in what were called internally generated movements as opposed to externally generated movements. And in fact, it was these observations by Okihide and Bob so many years ago that led to the models put forward by Roger Albin in particular about how Parkinson's and Huntington's disease might result from changes in this level of inhibition, the disinhibitory model for the aficionados. And so we were really interested in this, we're still interested in it, but a long time ago now we did an experiment to assess the hypothesis that these neurons were preferentially involved in memory-guided eye movements. And so here's just an example showing a series of eye movements made from a monkey, and the blue show you with stimulation and the black without stimulation. And we electrically stimulated the substantia nigra pars reticulata to change the activity of the neurons. And what we found is when the monkey made visually guided eye movements, they were virtually unimpaired. However, when the monkey tried to make memory guided eye movements, we saw profound impairment. In many cases, the animal could not make an eye movement to the location that was represented by the underlying neurons. And the important thing about this was that with electrical stimulation, of course, today we would probably try to do this with optogenetics, although that still is very hard to do in macaques. But we could change when the electrical stimulation occurred in time. And what we did is we put the electrical stimulation not only during the movement, but well before the movement, during that delay period, while the monkey was waiting to be told, okay, it's okay to make the saccade now. And we still got these changes in eye movements, even though they occurred much later than the stimulation. And so what this led us to think about was that the availability of visual information really changes the contribution of the basal ganglia to movement and to the events that precede the movement, like memory or specification of a saccade or decision-making. And because I've had a strong interest in Parkinson's for a very long time, I've always had this eye toward clinical phenomena, even though I'm not a clinician, it reminded me of a symptom that's known in Parkinson's called paradoxical movement. So people with PD, you may know someone, they have a very characteristic sort of stooped posture and a shuffling pattern to their gait. But if you draw high contrast horizontal lines on the floor and ask them to step over those lines, you can increase their stride length. So what that tells us is that the machinery, the locomotor machinery in the spinal cord is intact. And when there's visual stimuli there to drive it, you can access it. But in the absence of that visual stimulus, you can't, they cannot. And so it turns out that the same kind of phenomenon can be seen in eye movements. It can be recapitulated in eye movements. And it's easy to imagine how activity of neurons like this could give rise to that kind of a symptom. And so what we were interested in is it possible that this phenomenon might actually extend to cognition? Is there such a thing as a paradoxical decision-making? And so what we did, here I'm showing you the kind of stimulus that we show to human participants now. These are called glass patterns after the name of the person who discovered them, Leon Glass. And we show these to people and we just ask them to report, do you see orientation? Is it tilted to the right or to the left or not? For those of you who are familiar with the random dot kinetograms that was used and developed and popularized by Tony Movshin, Ken Britton, Mike Shadlin, Bill Newsom, you can think of these as sort of the form equivalent of those motion stimuli. Although these don't have explicit motion in them, people often do report that they see motion, so don't worry if you do. So when you show these to people and you ask them to tell you whether it's tilted to the right or to the left, you see that they will report when it's very clear, 100% certainty, so all of those dot pairs are correlated, so you can see it tilt to the right, they choose positive or rightward all the time. When it's to the left, they choose rightward never. And all of the choices in between, reflecting the different coherences that provide some level of difficulty to the decision. And what you see here is when there's only 0% coherence, that is that panel on the left where there's just noise, the participant guesses just left or right because they can't presumably see what the orientation is because there is no orientation. Now, a very talented postdoc in the lab, Alessandra Perugini, together with Jochen Diederich from Davis, we did an experiment with people where we wanted to manipulate the memory information and ask whether there was such a thing as paradoxical decision-making. And so what she did was she showed people these glass patterns, but she varied the color of them. And in so doing, she could then manipulate the probability that either the left or right would occur more often. But she did that only on one of the colored stimuli. So for example, the red one might have left and right. Left would occur 80% of the time, right would occur 20% of the time, and in the green one, they would occur 50-50. And she would randomly interleave all these trial types, and measure the decision-making performance, as well as the ability of the participants to use that statistical structure, that statistical information that they learned and remembered over the course of the experiment to guide their decisions. And when you do this in healthy people, what you see, in fact, is that they do use that information. So if the positive or rightward orientation is shown more often, they choose rightward or positive more often. If the left is shown more often, they choose left more often. But interestingly, people with Parkinson's disease show impairments in this. They're not able to properly use that prior probability information to guide or to exhibit a decision bias. So suggesting that there is a kind of paradoxical decision making. And if you look at the age and sex-matched healthy participants, usually the spouses of the people who have Parkinson's, you see that in fact they do use that prior probability information quite well. In fact, in some cases, they often overuse it. And this we know, in fact, from the economics literature, that as we age, we tend to rely on our heuristics, our gut, a little bit more, even in the face of data that might tell us something different. So we saw that in our patients as well. Now, a very talented grad student in the lab, Waibo Thakur, carried on this project, did some other work with humans that I won't talk about today, but he, together with our colleagues Barbara Knowlton and Leila Montasir-Kiosi, looked at patients, more patients with Parkinson's, and really wanted to get at what the role of medication was. Because that first data set that I showed you, those patients were all on their dopaminergic medication. But what Waibo found was that if he included more people, looked at on and off their medication, there was lots of variability. Some people were able to use the prior probability while they were on their medication, and certainly not when they were off their medication. Other people showed the opposite. So he sorted this out, and what he discovered was that it appears to depend on the particular phenotype of Parkinson's. So for those of you who know something about Parkinson's, you know that it comes in a variety of forms. It's not a uniform disease. Well, the disease process likely is, but the expression of the disease process is not. Some people show more tremor, other people show more bradykinesia, and it turns out that that matters. So the people who were tremor dominant saw benefit from the dopamine, but the people who were bradykinetic dominant did not. And so the influence of dopamine really depends, and their ability to use priors, and that influence depends on the phenotype. So to summarize this, people with Parkinson's show impaired integration of this sensory and memory information for decisions. There is a kind of paradoxical decision-making. And other people have shown similar results. Conrad Kurting comes to mind in his work, where there is this, seems to be this hyper-dependence on visual information in people with PD, using a different task. And the influence of dopamine really reveals underlying differences in decision circuits across patient populations. And one of the things we became interested in, of course, is what is the mechanism of this underlying in Parkinson's. And for this, we turned to some of the well-known models of decision-making, the sequential sampling models, a famous one being the drift diffusion model developed by Roger Radcliffe. And I just want to make a point here that this model and its application to neuroscience was really popularized by Mike Shadlin and his colleagues. And it really is because of that fundamental science in non-human primates that we even thought to do this in humans. And it's really important to remember that basic research involving monkeys, whether you see it a direct translation or not, it can give rise to useful information that's relevant for patients. And this is a very good example, in my opinion. So a shout out to Mike Shadlin. So this model, though, is very simple. You can imagine two choices, left or right, and then a starting point. When you don't have a bias, you start right without a bias. You start right in the middle between these two bounds. And then you have sensory evidence that you accumulate over time until you reach a bound. And when you cross that bound, you make your decision, choice one or choice two. And these are various parameters of this relatively simple model: the start point, the decision boundary, and this is called the drift rate. And so what we did was ask, well, in this model framework, how are biases implemented? And it's pretty straightforward. You can do this in two ways in this model framework. One is you can change your start point. You can bring it closer to the decision bound that is the more frequent orientation. Therefore, you have to accumulate less information to make your choice. That's one way to create a bias. And the other way is to provide a sort of additive gain almost to the drift rate itself. So an average coherence signal added to all of the coherences would give you a decreased threshold, in other words, reduced amount of evidence that you need to accumulate to make a choice. So this is called the drift rate offset. This is in the parlance of Roger Radcliffe's modeling. And so what, together with Jochen and also Waibo Thakur in the lab, we found was that healthy humans actually, to perform this task, they use both of those parameters to adjust their decisions. So they do a general start point change, and they apply that to both colors of the stimulus. And then to get the specific bias for that particular colored stimulus, they adjust their drift rate offset. Okay? And it turned out that people with Parkinson's could adjust their drift rate offset appropriately, giving us some suggestion that they're learning about the priors, that the brain knows something about the priors, but they were unable to adjust their starting point. So that's what led to their inability to apply that bias appropriately. And so we, again, wanted to understand how this came about. And so here's now, we go back to the non-human primate model, the monkey model, to understand the circuits. And what we did was, we know that Parkinson's symptoms are thought to arise from an increase or a change in the inhibition coming out of the basal ganglia onto the superior colliculus. So we did the simple, not so simple, experiment of putting mucimol, a GABA agonist, into the superior colliculus while monkeys performed these simple two-choice decisions. And I should point out these two folks here, Liz June and Alex Bautista, they did this experiment while they were post-bacs in the lab. Pretty amazing students. They're now both graduate students in neuroscience. And what they did was they had monkeys perform this task. They saw the glass patterns. If they saw it to the right, they made an eye movement to the right. If they saw it to the left, they made an eye movement to the left. And then they injected mucimol. Simultaneously recorded the raw voltage signals here, so you can see that the mucimol depressed the activity of the neurons. So basically shutting off the superior colliculus a bit. And when they did this, they found profound changes in decision-making performance. It introduced a bias, basically. And so the inhibition of the colliculus produced that ipsilateral, same-side decision bias. And you can see that in the orange traces here. Black is, of course, pre-mucimol. And then here is just showing you 6 injections in a couple of monkeys, just saline. And you can see that the saline had no effect. So there's a significant change in decision-making performance, an expression of a bias when you inactivate one superior colliculus. And in fact, Guidon Felsen and his colleagues have shown similar results in the rodent model with similar tasks, olfactory decision-making tasks. And importantly, what Liz and Alex also did was they had monkeys make simple eye movements in a simple visually guided task, but also a simple selection task where the fovea, there was a central target and it changed colors, and that told the monkey which target it had to look at, the right or the left. And when they did that, you can see in panel B and C, there was no change in the accuracy of the eye movements. So it wasn't just a general eye movement bias, that they were just looking at those other targets more often or less often. We really only saw the change in the decision task. So this told us that it really was a decision-making impairment that we were seeing and not just a motor or attention, actually, phenomenon. And then Liz, together with a former postdoc in the lab, Michael Nunez, did a series of modeling exercises. Of course, as I always say, we choose the best model suited to answer the scientific question that we're interested in. And if we need a monkey, we use a monkey. If we need a human, we can use a human. If we need a computer, we use a computer. And here's a case where we've used a computer to simulate the predictions of this model. And we took, well, not we, they took this traditional DDM model, this traditional sequential sampling model, and then simulated what would happen if we inactivated one parameter. And in this case, they're inactivating the parameter that is the drift rate. So what happens if what the colliculus is doing, inhibiting it, is making the sensory evidence less clear for the subject? And so this is just a simulation, so these are reaction time distributions. This is what you'd expect to see from the psychometric function, and then this is what you'd expect to see if you look at the distribution of reaction times. Now with this model, I won't go into the details here, but the important thing is not just the psychometric function, but it really is looking at the distribution of reaction time, and that's what tells you what parameter is being affected. And so then what they did was inactivated the start point. What happens if we just change the start point or remove the start point? What is the prediction? And you see now you get a shift rather than a change in slope, as you see with the drift rate. And you get these very predictable changes in the reaction time distributions for correct and incorrect choices. And then finally, if it's the drift rate offset that's changing, what we see here is you get a nice shift in the psychometric function and these changes in the reaction time. Importantly, the incorrect reaction times aren't changed all that much. So then we just did the statistical test that my PhD mentor told us, taught me rather. He called it the interocular trauma test. We just looked at the data and we said, what do the data look like compared to those simulations? And when you do that, these are the examples now, or actually the raw data from the two animals. And you can see that there's a nice shift in the psychometric function here. There's not a change in the slope, so we can rule out the drift rate. And if you look at the reaction time distributions, correct and the incorrect, they're kind of noisy, but on average, on the whole, it's much more consistent with the predictions for the drift rate offset than it is for any other one of the parameters. So the inhibition of the colliculus changes the starting point a little, but mostly it's the drift rate offset. And Michael and Liz did a really exhaustive modeling exercise. If you're very interested, I'd encourage you to take a look at the paper as well. So this, in summary, an interim conclusion here is that the inhibition of the colliculus really behaves as though we're subtracting a coherence-independent constant from the evidence signal for contralateral decisions. It's basically shifting the balance of evidence away from the inactivated field. And this is important because what it means now is the colliculus is actually participating in the computation of decision. We don't have evidence that it is participating in the accumulation process itself. It may be, but these experiments don't speak to that. But what they do speak to is that the colliculus is participating in the computation of decision evidence in some way. And what we think is that it's actually providing a gain modulation, and in this case, an additive one. Importantly, though, mucimol is not specific. When we put mucimol into the colliculus, we affect many different neuron types in the colliculus, and we affect many different regions of the brain because the colliculus has projections virtually to the entire neuraxis through its ascending pathways, through the pulvinar and the thalamus, and through its descending pathways to the brainstem. So when we put mucimol in there, we're affecting a lot of cell types and a lot of circuits. And so what we want to do in the monkey, of course, is we want to get some specificity. We want to dissect the circuits. And so our collaborators, Alev Irishir and her grad student, Arda Kupchak from the University of Virginia, have developed a new promoter from the tree shrew genome that targets specifically neurons in the colliculus that are projecting just to the pulvinar. And they did injections in the tree shrew and the mouse, and a beautiful paper just came out in Cell Reports, or it was just accepted rather. And then in collaboration, another very talented grad student in the lab and talented former postdoc in the lab, Chris, and in collaboration with Martin Bolin, our colleague from Duke University, they made injections into the macaque monkey. And this is the data from Alev and Arda now in the tree shrew. What you can see is that this promoter is actually quite specific for the lower superficial layers of the colliculus, targeting those wide field vertical neurons. That's what they're called. These are the neurons that we know project up to the pulvinar. Okay? And then when we make the injection in the macaque, you can see that we label them also very nicely in the macaque. And you can see the layering here highlights that lower superficial areas in the macaque. There's some labeling here of the intermediate layer neurons, and it turns out that it depends on the titer, to my surprise. I don't fully understand the biology there, but when the titer is very high, it loses its specificity. So we're working on trying to get these titers just right. And a shout out to also our colleague at Washington, Greg Horowitz, who helps us a lot with these things. And so in the meantime, we're also very interested in how this works in mice. And so what we can do is we can take advantage of mouse models, the transgenic tools, to reversibly inactivate these cell types specifically in this animal model. And so there are Cre lines of mice, this NTSR one that expresses Cre exclusively in those wide field vertical neurons. And then what we can do is inject the receptor and an HM4D receptor, which is a chemogenetic or DREAD receptor that inactivates neurons, into the colliculus to specifically label those cells that project up to the pulvinar. Or what we're trying to do now is use the promoter that Alev and Arda developed. But talented grad student Waibo Thakur developed this task in mice. And so he's been able to train mice to do this motion task now, this random dot motion task, where mice have to observe this screen and make a decision about whether or not the dots are drifting to the right or drifting to the left. And you see the mouses moving the wheel, but the choice report, the perceptual report, is actually in the lick. So the mouse has to lick to the right if he sees it going temporally, and he has to lick to the left if he sees the motion going nasalward or rostrally leftward. Yeah, left and right. But the mouse uses the wheel too, but it's interesting because it looks like the wheel sometimes goes in the opposite direction that the mouse is licking. So that's a whole rich data set that we have yet to look at. But importantly, what Waibo's been able to do is train these mice to do this task, and he gets very nice psychometric functions from mice. They're not as great as monkeys, as those of us who work with both species know, but they can do the task. And he had a really nice poster on this the other day. I hope some of you may have seen it. But what he also showed was that the mice actually engage in a bounded accumulation type strategy to solve this task, to do this task, just like is seen in monkeys. And these are data now from Ruz Bekiani and Mike Shadlin. And this is sort of the hallmark of this bounded accumulation kind of model, where as the coherence increases, the ease of the decision increases, you see this type of less dependence on the stimulus, or dependence on the stimulus duration. And if you look at this in mice, you see a very similar kind of scaling with coherence. Again, it's not as pretty as you see in monkeys, but it's still there, suggesting that they do use this accumulation type model. And what Waibo also did was he introduced just pulses of motion evidence to see whether and when, during the trial, the animal would use that additional motion information. And you see that when he provided the pulse early or middle, the mouse used that information. But if you did it later, they didn't. They didn't use it as much. And again, this is consistent with the kind of bounded accumulation model of decision making. And then when he inactivated these wide field vertical neurons, these are very preliminary data still, but it's consistent, he again saw the bias here. And so what we conclude from this is that these pulvinar projecting wide field vertical neurons are sufficient to produce a bias in decision making in the mice. And our hope is that we can see this in the monkey soon. So the important thing also to show you that Waibo did was he showed that this was not dependent on, or was not a purely motor phenomenon. So if you just looked at the distribution of licks during the inactivation, there was no significant difference. There were, a lot of variability here, of course, they're mice, but there was no significant difference. And also, it was not dependent, it was not a purely visual thing. It's not like the mice just were blind, because if he blocked the motion stimulus by just putting a barrier between the eye and the screen, the effect went away. So you see it just goes away here, but when the mouse's eye is available to see the visual information, he sees a change in the choices. So it's neither purely motor nor purely visual. It's a bona fide decision deficit. And so the novel hypothesis here is really that we focus so much on the descending pathway of the colliculus, but in fact the ascending pathways are relevant for behavior, and likely it's feedback from the colliculus that may regulate how decision evidence is accumulated for decisions. And so, okay, what's happening now? There's a lot of really lovely work in other species. From Herwig-Bayer and also in the zebrafish, and the group in China, Zhang and his colleagues, using transcriptomics to look at evolution of cell types. And in my view, this is a really fantastic direction. And a really interesting finding from the Zhang group recently is that they identified in the avian tectum a cell type that is very specific to the avian tectum. If they look at a mouse, they find, molecularly anyway, if they look at the mouse, that same cell type, the molecular structure, is found in the cortex in the mouse. It's not in the colliculus, as it is in the bird. So really interesting kinds of questions, I think, can be asked. And one of the ways that we're trying to sort of get at these ideas is to look at ex vivo slices of monkey and compare those, of course, with mice. And there's very lovely work done by Gabe Murphy and his colleagues, classifying neurons using physiological and morphological parameters. And we started this collaboration with our colleagues, Dr. Martha Bickford and Dr. Sean Masterson at Kentucky, and also Nick Dembro in Washington. But Travis here, a really outstanding biophysicist, has done this work that I'm showing you now. And what he did was he recorded from slices of the colliculus, both in mice and in monkeys, and he characterized the cells electrophysiologically and morphologically, and then used a dimensionality reduction procedure to cluster the cells, to try to identify common or different characteristics. And you can see here that some overlap between mice and monkeys, and you can see the activity of really high firing rates, a SAG that is very common when and mediated by HCN channels, for example. And then this other type, they're a little bit different between mice and monkeys, shown here. So this is one approach. And looking at the physiological features, you can see that some are not different between mouse and monkey, but some are quite different. So the action potential width, the threshold of action potentials, the latency to the first spike. So looking at these properties of these cell types and comparing them across species, and then next step, doing single cell or single nucleus RNA transcriptomics will help us identify the conserved and divergent features across animal models and species, giving us some clue into the evolution of vision and cognition. And then finally, the sort of last step that we're taking is trying to put all this together now and look at this in behaving monkeys. And this another exceptional graduate student, Mary Hong, together with Weibo and our colleague Edgar Walker, looking at monkeys performing our choice task with priors and without priors. And you can see that monkeys use priors just like humans do. And you see statistically significant changes in the shift, just like we saw in humans. And so doing this, Mary was then recording from the colliculus to see how is this memory or prior information encoded in superior colliculus neurons, and how do collicular neurons perhaps integrate this sensory and memory information to guide the choices of monkeys in this task. And what you can see is that there's a lot of heterogeneity in the colliculus. We usually think of the colliculus as they're visual cells, they're visual motor cells, they're motor cells, they all act the same, and we average them together and look at these spike density functions that show you the average activity. But in fact, if you dig into this, you see that Even collicular activity is quite heterogeneous. We do see evidence for scaling with the coherence, suggestive of an accumulation process, and we also see changes in the gain of activity with unequal prior compared to equal prior. So there's a mixed kind of evidence in these cells across different cell types. And so trying to move away from the drift diffusion model, which sort of relies on similar neurons behaving similarly, we turned to more recent advances and the GLM-HMM model, general linearized model with hidden Markov model. And I'm sorry I don't have the citation here. This is from the Jonathan Pillow and Ann Churchland, and the IBL did this in mice. And we just sort of built on that to look at this in monkeys. And the idea here is, because most classical models of decision making assume that there's really only one consistent strategy throughout the experiment. But these GLM-HMM models allow you to look at the behavior and see whether there are actually latent states that are driving the behavior. Because as we know, it's probably unrealistic that subjects are not doing different things, even across an individual trial. And so what they showed in IBL is that mice actually have a few states when they're engaging in a simple decision task, and they kind of vacillate between these states throughout a trial. And so Mary and Weibo, together with Edgar, applied this to the behavioral data in monkeys and showed that, in fact, they only show two states. They're much more consistent than mice seem to be. And in the unequal prior trials, you get this nice biased state. And in the equal prior trials, you see a very nice stimulus or coherence-dependent state. And then just training the model versus looking at the test data set. You can see the test data set is increasing here, probably resulting from overfitting. So we concluded really that this two-state model was the best for behavior. And if you look at this transition matrix, you see that monkeys' behavior is quite sticky. You know, when they're in one strategy, they sort of stay in that strategy, which is interesting and different from mice. But what's nice about this model is now you can recategorize or reassign your trials based on these latent states. So you're now away from the experimenter imposed, this was a prior trial, equal prior trial, unequal prior trial. Now you can sort the trials based on the state that your model told you that the animal is in and look at the activity. And so they did this in combination with demixed principal components, analysis, which is a technique developed by Christian Machens, and then you take the neuronal activity sorted this way and project it back onto those latent states, and what you see is that the colliculus has a very nice representation of the bias of the prior information. So you get big differences here between the equal and unequal prior states. There's also evidence for coherence. So the stimulus information is also contained within in the collicular activity. And then also, of course, the choice. The solid and the dashed indicate choices to the response field of the collicular neuron, and the dashed is away. And so when they looked at this across the whole population, what they found was quite a bit of selectivity and mixed selectivity. So superior colliculus is looking a little bit more like prefrontal cortex, we think about, than the traditional way that we think about the superior colliculus. So what we want to do now is combine these new analytical tools with transcriptomics, ex vivo, in vivo work to really dissect these circuits. And so, in conclusion, despite its small size, we think the colliculus is very cool, and the ascending circuits are likely to be very important as we think about the evolution of visual cognitive behaviors and depend on cerebral cortical areas that are absent in small mammals and birds. And then combining these tools together, we think is going to give us a lot of new and exciting information about the role of the colliculus in evolution of cognition. So, thank you. I think I went a little over. Not too bad.
Thank you, Michelle, for that just amazing talk. So we do have some questions that I'll share. First, I just want to share that there's a lot of love for the intraocular trauma test. So thank you for sharing that with us.
That's Craig Evinger, my PhD mentor, yeah.
Fantastic. So first question is, in comparisons with patients being on or off dopamine medication, how would your results potentially change with patients with DBS in terms of that accumulation?
Yeah, thank you. Thank you for that question. It's something I've been wanting to do for a very long time, actually, is to look at that. It's really hard to do, though, because the people with DBS are often quite debilitated. So when you turn the stimulator off, they have a really hard time even participating in the task. So it's been difficult for us to collect those data. One of the things I can say that we're interested in is there are different PD phenotypes, as I mentioned, and so neurosurgeons will target different regions of the basal ganglia. So in some cases, they'll target the subthalamic nucleus. In other cases, they will target the globus pallidus. And so what we'd really love to do is see whether we get different effects on the ability to use priors, depending on whether it's the STN that's being stimulated or the GPI that's being stimulated or GPE that's being stimulated. And we just haven't done those experiments, but we'd love to.
Great. So another question now for the more recent section, the GLM-HMM. Did you see any state-dependent differences in trial-to-trial spike count variability?
Yeah, so we haven't looked at that yet. This is really hot off the press stuff. My expectation will be yes, but we haven't looked at that yet. Weibo, Mary, are you there? Make a list.
So another question that I was wondering if you could maybe speculate on, you've looked, just beautiful work looking at a particular kind of decision where you're accumulating evidence. Given what you're seeing now with this hint at maybe some mixed selectivity sorts of things, can you speculate on maybe what other kinds of decisions the superior colliculus might be involved in and what you might see in that sense?
Yeah, so I guess there's a thinking that the colliculus would be relevant only for decisions that involve a choice report that is provided with an eye movement. But there's very lovely work from Rob McPeak's lab in a simpler task in the sense of its target selection. I won't go into the details, but he shows that if you, and his group show that if you inactivate the colliculus and the choice report is with a joystick arm movement, you still see the same kind of impairment. So my hunch is that it's something more broad than just eye movements, but that is an empirical question that we have not yet asked. There's also the lovely work of Rich Krausless that I didn't talk about. This is more in the change detection, attention field, but he sees similar things, and his group sees similar things when you inactivate the colliculus and have the animal report a change in motion direction with a joystick. So I suspect it's a more generalized phenomenon, but that's empirical, and we haven't done that yet.
Very cool, thanks.
Mm-hmm.
So it was striking the amount of overlap in the cell types between the monkeys and the mice, and also that there seemed to be almost an equal amount of divergence in the cell types. And I was wondering if you have thoughts about what could be different there, or what's the same, what those might be driving.
Yeah, and thank you for that question. I have no idea. But, it's fun to think about, right? They have to be doing something different because primates don't rely on these cells to see in the same way that other species do. But they're still there, and they're still doing something. They have the same projection patterns. So trying to tease out what that is and using all the tools that we now have available to us to look at that, I think is fascinating. Yeah, I don't know. I don't know.
Well, we'll look forward to more. And with that, help me in thanking Dr. Basso again for just a wonderful talk.
Thank you. Thanks for sticking it out till the end. I really appreciate you being here.